Hello there, welcome to Fundamentals of Biology, where in this session we're going to be looking at osmoregulation and excretion. So by the end of this session, you should be able to describe the role of osmoregulation in homeostasis. You should be able to discuss the osmoregulatory challenges faced by animals living in different habitats and how they are met. Should be able to describe the role of excretion in osmoregulation, and then describe the structure and function of vertebrate kidneys. So there's quite a bit to get through in this session, but we'll work through it, and then as always at the end I'll have some test questions for you to see how you've gotten on. Physiological systems of animals operate in a fluid environment. And what does that mean? Well, it means that animals contain large quantities of water. So as adult humans, we're made up of somewhere around 50 to 60% water. Different species will be made up of different amounts of water, but we also need liquid water to survive. So not only do we contain a lot of water, we're constantly interacting with water. We need to consume it in our food, and we consume it obviously as liquid water as well. We lose water through evaporation, through urination. So there's this constant intake and excretion of or loss of water, and we need to maintain the correct or the right amount of water within our bodies to keep everything functioning correctly. So relative concentrations of water and solutes. So if you remember from previous sessions, we've talked about the fact that water is an excellent solvent. That means that things can dissolve readily in it. So we end up with lots of solutes, so sugars and salts and amino acids, things along those lines. Well, they can dissolve readily in water. So when we're talking about the insides of animals, we're not talking about pure water. We're talking about water that contains a lot of different solutes. And the balance of water and solutes need to be maintained within fairly narrow limits. Again, we've discussed this in a previous session where we looked at cell membranes and we talked about osmosis. So hopefully you can remember that with osmosis, it is the diffusion of water from an area of low solute concentration to an area of high solute concentration. So water will diffuse into an area that has a higher solute concentration. So you can imagine that if your blood, for instance, had a higher solute concentration than the inside of some other body cells, well, that will cause water to leave those cells via osmosis. And as we've established in that session, that will cause the cells to shrivel up and ultimately die. So it's incredibly important that bodies are able to regulate or maintain their water and solute balance within these fairly narrow limits that enable life to exist. That process of balancing water and solutes is what we refer to as osmoregulation. So osmoregulation controls solute concentrations and balances water gain and loss. So why have I got a photo of an albatross on this slide? Well, albatross live very, very difficult lives in relation to osmoregulation. They only come in contact with seawater, pretty much. They rarely have access to any large quantities of fresh water. Obviously, they'll encounter it when it rains, and they may get some standing fresh water on the occasions that they return to land. But typically, their life is surrounded by or spent surrounded by salt water. So they have to work very hard to osmoregulate so they don't end up taking in too many solutes and losing too much fresh water. And we'll talk about some of the ways in which they do that as we go through. Different animals that live in different habitats and different environments will face different osmoregulatory pressures. So as we can see here, desert and marine animals face desiccating environments that can quickly deplete body water. So we can see the Arabian oryx here. Well, they live in a very dry habitat, so they are constantly at risk of losing water through evaporation, and they don't have access to lots of standing fresh water that they can replenish that water with. So they are constantly at risk of drying out, or what we refer to as desiccating or desiccation. On the other picture there, we have clownfish, which are marine fish, and they live in a hypertonic environment. So seawater is hypertonic relative to the inside of the fish. That means that seawater has a higher solute concentration in relation to the tissues of the fish. That means that they are constantly at risk of losing water via osmosis. And we'll look at how they combat this as we go through. Freshwater animals, so if we, instead of looking at a clownfish, if we were looking at, say, a freshwater trout, they will have the opposite problem. Because now the inside of the fish is actually hypertonic in relation to the water they're living in, so they have a higher solute concentration in their tissues, they are constantly at risk of gaining too much water through osmosis, but also losing solutes via diffusion. Alongside of this interaction with water in the environment around you, you also have the issue surrounding the buildup of other solutes and particularly waste products. So as animals, we consume food, and that food goes into producing energy through respiration. So we're generating ATP, and we're also building our bodies, and we're repairing them. We're producing gametes, so eggs and sperm. There are a lot of processes going on in our bodies, and they generate quite a lot of waste products. Particularly when we break down things like proteins and nucleic acids, we get the buildup of nitrogenous wastes. And these have to be dealt with. You have to excrete them, because otherwise they will just build up to dangerous levels within the body. So excretion rids the body of nitrogenous metabolites and other waste products. So let's just do a quick recap of osmosis then and osmolarity, and then we can start to put it into context. We can start thinking about it in terms of osmoregulation. So we know that water will enter and leave a cell by osmosis, and that osmolarity is the solute concentration of a solution. And that osmolarity or solute concentration will determine the movement of water across a selectively permeable membrane. So remember, water will diffuse from an area of low solute concentration to an area of high solute concentration. So water will always diffuse towards the higher solute concentration. If, however, you have two solutions that have the same solute concentration on either side of a membrane, we would refer to that as isoosmotic. Sometimes you'll see it referred to as isotonic. And this means that water molecules are balanced there. The amount of water molecules and solutes are the same on either side. So you will get the movement of water molecules across the membrane, but this will be at equal rates, and it will be in both directions. So you'll have no diffusion of water from one side to the other. And if you remember from the session that we did on cell membranes and membrane processes, this is the condition that animal cells like to be in. So we want our blood and our tissues and our cells to be isotonic with each other. If two solutions differ in osmolarity, the net flow of water, so the diffusion of water, will be from the hypoosmotic, so hypo meaning lower or less than. So this is the lower solute concentration, to the hyperosmotic, so hyper meaning higher or more than. So the hyperosmotic or more concentrated solution. So water will diffuse from a hypoosmotic to a hyperosmotic area. Keep those terms in mind because they'll become important as we work our way through. So when it comes to osmoregulation and animals then, we first can break it down into, or we can put our animals into two different groups. The first group is quite a rare group, and they are referred to as osmoconformers. And as you could probably imagine with a name like that, these are animals that do not invest any energy in osmoregulating or maintaining an internal osmolarity different to the environment in which they live. So osmoconformers, consisting only of some marine animals, are isoosmotic with their surroundings. So the internal or the inside of these animals will just be the same as the water in which they're living. So they do not regulate their osmolarity. The benefits of that, well, they don't have to invest any ATP. They don't have to invest any energy in osmoregulating. The downsides to that is that it does mean they have to stay within some fairly narrow limits in terms of the osmolarity of their environment. A quick thing to point out here is this bit here that some people get confused about when they first learn this topic. So consisting only of some marine animals. Okay, this doesn't mean that all marine animals are osmoconformers. Far from it. The vast majority of marine animals are osmoregulators, but what it does mean that all osmoconformers you will find in the marine environment, and they are restricted to just a small group of marine invertebrates, such as the cnidarians. So we can see this jellyfish here. Okay, but most animals are osmoregulators, so they will expend energy to control water uptake and loss in a hyperosmotic or a hypoosmotic environment. And the reason I put these salmon here is because salmon, as you probably are aware, they are born in fresh water. So when they are in fresh water, they are living in a hypoosmotic environment. So they have to osmoregulate appropriately for living in a hypoosmotic environment. And then they will go through a process of smolting, which is where they prepare to move from the freshwater habitat into the sea, and then they will be living in a hyperosmotic environment. So they go from freshwater to seawater. And to do that, they have to go through really large physiological changes to prepare their bodies for that change in osmoregulation. Osmoregulation is where animals will expend energy to regulate or maintain an osmolarity that's different from the external environment. Osmoconformers, they don't spend that energy. Their internal osmolarity will just be the same as the environment they're living in. And when we look at aquatic animals particularly, what we find is that most of them fall into a group that we refer to as stenohaline. And this means that they cannot tolerate substantial changes in external osmolarity or solute concentration or salinity, depending on how you want to look at it. Okay, so most animals they have to live in a fairly narrow limit. So seawater fish, for instance, well, they need to live in the sea. If you take a cod from the sea and you plonk it into a freshwater pond, well, it's going to die because it's not it's a stenohaline animal. It's not able to tolerate the changing salinity. And the example I've put on here are the bluefin tuna. So these will not survive in fresh water, certainly not for any length of time anyway. Although we do get some aquatic animals that are euryhaline, and these are animals that can alter and adjust their osmoregulatory mechanisms, and we'll look at some of these in a moment. They are able to adjust and make changes to the osmoregulatory mechanisms, which will enable them to move from freshwater into seawater, seawater into freshwater, some for very long periods of time, some for just short periods of time. And essentially they can change and survive these large fluctuations in external osmolarity. One of the most famous examples of this that you can see on the right here is the bull shark. So bull sharks are, they live in the sea, so they're marine animals, but they are well known for being able to move from marine habitats into estuaries where the water is brackish, so a mixture of fresh and salt water, and then right up into river systems where it's fresh water. And they can stay up there and for quite a long time and travel long distances upriver. But most animals are stenohaline. They cannot tolerate these changes in external osmolarity. But just be aware that there are some that are able to do this, and we refer to those as euryhaline. And we see quite a few species of these in the UK. So if you go to any estuary in the UK and look at the fish that you see in there, so common examples would be fish like grey thick-lipped mullet. You might find some flounder, sand smelts, sand gobies. So you'll get a lot of fish like bass, sea bass, for instance. They will move from the sea and they will come into estuaries to feed or to breed. and the salinity in those estuaries will often be much, much lower than what you would expect to see out at sea. So let's start by discussing marine animals then. And as we talked about previously, most marine invertebrates are osmoconformers, so they don't invest any energy in osmoregulating. However, all marine vertebrates are osmoregulators, so they need to invest quite a considerable amount of energy in maintaining a different osmolarity internally to the seawater they're living in. And this is because marine bony fish are hypoosmotic to seawater, so that means their internal solute concentration is lower than seawater. And how do they overcome this? Well, they balance the water loss, so they're losing water through osmosis, and they will counter this by drinking large amounts of seawater, and then they are relying heavily on their kidneys to eliminate the ingested salts through the kidneys in highly concentrated urine. And then they also have specialized cells in their gills to actively transport salts from their blood to the surrounding seawater. So again, if you go back to that lesson that we did on cell membranes and membrane processes, active transport is the movement of substances from an area of low concentration to an area of high concentration, so against the concentration gradient. And to do that, you need to expend or use ATP. So they're actually having to spend energy to pump salts across the gills into the surrounding seawater. And they're relying on their kidneys to produce very concentrated urine. And then here we can see a sperm whale, and we see that with marine mammals, they have highly adapted kidneys that enable them to filter out these salts and excrete them in their urine. And here we can see a diagram that shows those processes within a marine fish. So we can look at the key here, and we can see the dark blue arrow is for salts and the light blue arrow is for water. Let's look at water first. So water is being ingested. So sea fish or marine fish they drink large amounts of water, but they are losing water constantly through osmosis. So osmotic water loss through the gills and other parts of body surface. And they are losing water through osmosis because water will diffuse from an area of low solute concentration, so the inside of the fish, to an area of high solute concentration, the seawater around them. So they're losing water through osmosis, they're gaining water in their food and also through ingesting it, so they're drinking it, and they lose some extra water through producing urine. But what they are doing is producing highly concentrated urine. So they're trying to conserve as much water as possible whilst getting rid of the excess salt. So we can look at the salts with the dark blue arrows now. So they are gaining salt ions in their food and also from the water they're drinking. They are losing salts in their excretion. So they are urinating out some salts that are filtered out from the kidneys. And they are using those specialized cells, so chloride cells in the gills, to actively transport salts from the blood to the surrounding water. So this is how a marine fish osmoregulates. There's quite a lot going on there, and it does require quite a bit of energy. So we'll look at how kidneys work later on, and we can see that there is an element of active transport within the kidneys. So you are relying on ATP and also for the chloride cells within the gills. So there's quite a bit of energy, and there's quite a lot to consider when we look at osmoregulation in marine fish. When we consider freshwater animals, well, they have the opposite problem to what we've just discussed with the marine fish. Freshwater animals are constantly taking in water by osmosis because they are living in a hypoosmotic environment. So they are gaining water through osmosis. They are losing salts through diffusion. So they lose salts by diffusion and maintain water balance by drinking almost no water and excreting large amounts of dilute urine. So we can see how that's the opposite of what we just talked about with marine fish. Freshwater fish, they don't drink water. They will take some in when they feed, but they are not actively drinking water, yet they are almost constantly creating quite large amounts of very, very dilute urine because they are trying to flush out water but hold on to the solutes. The salts that they lose through diffusion and in their urine, they are replacing in their food, so they are getting solutes in their foods, but they also have the same specialized cells in their gills that marine fish have, but they just work in the opposite direction. So whereas marine fish, they use active transport to get rid of salts, well, freshwater fish do the opposite. So they use active transport to pump salts from the water into their bodies. So they're still using active transport, they're just doing it in the opposite direction. And here we can see the same diagram that we looked at previously, but now for freshwater. So again, dark blue arrow is for salts, light blue is for water. So they are going to gain some water in their food and when they feed, but they're not actively drinking it. They are constantly gaining water via osmosis. And you can see here by the width of this arrow that they are getting rid of a lot of water through their urine. So excretion of salt ions and large amounts of water in dilute urine from the kidneys. And then with the salts, they're gaining some salts in their food. They are having to pump some solutes or salts from the water across their gills. And unfortunately for them, they will also be losing some salts in their urine. Because when we look at the kidneys, we'll see that the filtration process in the kidneys is non-selective. So you're always going to lose some solutes in your urine. So we can see here that freshwater fish does things pretty much in reverse, or the opposite of what we see in the marine environment, which always, it never fails to amaze me really when you look at something like a salmon that is able to move from freshwater to saltwater and then back to freshwater, or you look at something like a mullet that is able to freely move from seawater to freshwater and back to seawater in the space of a few hours if it wants to. And so to be able to do that, they have to change the way in which they osmoregulate, which is pretty impressive. When we start looking at land animals, we need to realize that it's very, very easy to lose water when you're a land animal or a terrestrial animal. And that is because water will very easily evaporate, particularly in hot and dry or arid environments. So you can lose an awful lot of water through evaporation. We're also producing urine, for instance, to get rid of our waste products. So it's very easy to lose water as a land animal. So we have to have adaptations to reduce that water loss. And the way in which we do this, well, some of them can be fairly straightforward. So the first thing is obviously we have body coverings. So our skin, for instance, is largely impermeable to water. If our skin was highly permeable to water, well, our water would very quickly evaporate out of our bodies and we would dry up and mummify, which wouldn't be very conducive to living a long and healthy life. So we have body coverings, we have skin. Different animals will have different types of body coverings as well as skin. So you'll see reptiles with scales, for instance, and birds will have feathers. So there's other body coverings, but ultimately the skin is going to be what helps them to prevent dehydration or to prevent them from desiccating. Obviously, this becomes a little bit different when we look at things like amphibians. So a lot of amphibians, their skin is largely permeable to water, so they have to stay within moist habitats to prevent them from drying out. We also see the same thing with some invertebrates as well. So if you are an animal that is at high risk of desiccating or drying out, well, then you are limited to staying within moist areas or areas where you can get access to water quite quickly. What we see with animals that live in extreme environments where there is a really high risk of drying out or desiccating, such as deserts, you can start to see all kinds of interesting adaptations. So you're not just relying on skin coverings, for instance. You need to go a little bit further. And some of these are fairly straightforward and quite obvious, but we don't always think about how they relate to something like osmoregulation. So we can get desert animals here, get major water savings from simple anatomical features like skin and scales, but also behaviors such as a nocturnal lifestyle. So we often see that with desert animals. They will adopt a nocturnal lifestyle, not only to avoid the incredibly hot temperatures during the day, but also to stay away from the increased risk of desiccating, of losing water through evaporation during the day. If you're out at night, not only are you not overheating, you're vastly reducing the amount of water that you're going to lose through evaporation. And land animals will maintain their water balance through eating moist food and also consuming water, and also producing water metabolically through cellular respiration. So in a previous session, we've looked at the process of cellular respiration, and we saw that water is produced during glycolysis and also through oxidative phosphorylation. So we are getting water produced during that process, and animals that live in extreme environments like deserts are particularly good at conserving and using their metabolic water, because obviously they don't have access to a lot of freestanding water that we do in living in a temperate environment in the UK. So if you're living in a desert, you'd better get good at using your metabolic water that's produced through cellular respiration. So how do animals actually carry out osmoregulation then? Well, we've talked a little bit about how some of the large mechanisms that we can use, but how does it actually work on a cellular level when we're starting to look at specific tissues? Well, animals regulate the solute content of body fluid that bathes their cells. So that's what we refer to as the interstitial fluid. So all of our cells are bathed in this fluid. And then we have transport epithelia. So if you remember, epithelial tissue that we've talked about in a previous session. Well, transport epithelia are epithelial cells that are specialized for moving solutes in specific directions, such as the active transport that we talked about previously with the fish, moving salts either in or out of the body. What we also find with transport epithelia is that they are typically arranged into a complex tubular network. And those complex networks of tubules are often arranged in a counter-current flow arrangement. And an example of this we can see in the nasal glands of marine birds, but we also see something similar in the animal that I had on the front slide of this lecture, which was the marine iguana. An example is the nasal glands of marine birds, which enable them to remove excess salt from the blood. And they use these counter-current exchangers to maintain concentration gradients that enable the solutes, the salts, to constantly move from the blood into a secretory fluid that then they can excrete from the body. So if we look at a diagram of an albatross, so I showed you a photo of an albatross earlier on and pointed out that one of the issues they have is that their life is just spent surrounded by seawater, essentially. They rarely come into contact with fresh water that they can actually drink. So they have these specialized nasal salt glands that enable them to remove those salts from their body. And you can see here this arrangement of complex tubules and then this secretory fluid that is being produced to help remove the salt from the blood. This is the important part of this diagram, really, because you can see that you've got the solutes, the salts in the blood here as these balls, and then you've got the secretory fluid that is being produced here. And notice that they flow in opposite directions. What this does is it helps to maintain a concentration gradient of the salt. So it's constantly diffusing down its concentration gradient from the blood into the secretory fluid, and then ultimately that fluid will exit at the top of the beak here. So you'll often see it look as if they have tears coming out of their eyes. Well, they're not coming out of their eyes. They're being excreted or secreted here at the top of the beak from these openings. And we see something similar with penguins, and we see something similar with, like I said, the marine iguana. So this countercurrent flow of secretory fluid and blood enables them to osmoregulate. This is how those seabirds are able to survive in a marine environment with very little access to fresh water. The next thing we need to start thinking about is the buildup of waste products within the body. So I mentioned this towards the beginning of the lecture. Not only are we exchanging solutes and water with our environment, we're also, as we consume food and carry out metabolism and growth and repair and mitosis and meiosis and ATP production, all of these processes of metabolism and growth and development that are going on, well, they produce waste products. And those waste products will build up over time, so they need to be dealt with. They need to be excreted or gotten rid of the body, because otherwise, as they continue to grow in concentration, they will affect the osmolarity of our tissues, and some of them are actually toxic as well, so will cause the breakdown or degradation of cells and tissues. An animal's nitrogenous waste reflect its phylogeny and its habitat. So let's go through and break down what that means. So the type and quantity of an animal's waste products may greatly affect its water balance. And as we'll see, it's not only does the buildup of nitrogenous waste particularly, but waste products, not only does it affect the osmolarity of the tissues, different types of nitrogenous waste will require different amounts of water to help the excretion of them, but also that will be determined by the habitat and lifestyle that those animals have. Among the most significant wastes are nitrogenous wastes, as I've mentioned previously, and these are a result of the breakdown of proteins and nucleic acids. And we've talked in a previous session very early on in this series about the fact that when we consume an excess of carbohydrates in our diet, our bodies are able to convert those carbohydrates into fats, into triglycerides that we can store within our adipose tissue, and then we can store that energy for later use. But unfortunately, when we consume excess proteins and nucleic acids, so nucleic acids, if you remember, DNA and RNA, so they are in all the cells that we consume in our food. When we consume these things, there's no real way of storing them or storing the excess for later use. We use what we need, and then we have to excrete anything that's left over. One of the big products of the breakdown of proteins and nucleic acids are nitrogen-based products. So if you remember, proteins are made up of amino acids. Amino acids have the amine groups or the amino groups that contain nitrogen, as do some of the R groups. And then nucleic acids have those nitrogenous bases that also contain nitrogen. So we get this buildup of nitrogen that has to be gotten rid of. And nitrogenous waste products come in three main types within animals. And we need to go through and have a look at each one of them and talk about how they are determined by the type of animal that we have, its anatomy, its physiology, and its habitat. So nitrogenous waste in the body tend to form toxic ammonia. So that's NH3, and that must be excreted. Some animals will convert ammonia to less toxic compounds prior to excretion. So the other two of our nitrogenous compounds are urea or uric acid. Okay, so these are our three main nitrogenous wastes that we deal with animals: ammonia, urea, and uric acid. So what we're going to do is go through and look at different types of animals and look at who excretes what, essentially. All three of those differ in toxicity and the energy costs of producing them. The first of our nitrogenous wastes to discuss is ammonia, as this is the first waste product that's produced by the metabolism or through the metabolism of proteins and nucleic acids. And ammonia comes with a whole host of issues. The most important one being that ammonia is incredibly toxic to life. So you cannot allow ammonia to come into contact with cells or body tissues for any duration, really, because it will just kill off those cells. So ammonia is incredibly toxic to life. One of the benefits of ammonia, though, or one of the good things about ammonia, is it's highly soluble in water. If you are an aquatic animal and you are constantly living in water, well, then it's very likely that ammonia will be your major waste product. So you can use that to get rid of your nitrogenous wastes. So animals that excrete nitrogenous waste as ammonia need access to lots of water. So obviously I've got examples here of coral and lobster and a little tompot blenny there, which is one of our native fish. And they are free to excrete their nitrogenous waste as ammonia. So they will often have specialized body surfaces that will enable them to get rid of this ammonia. So they release ammonia across the whole body surface or specialized areas. So if we're looking at things like corals, well, they can excrete or let ammonia diffuse across their whole tissues. Or if you're looking at something like the tompot blenny over here, the majority of their nitrogenous waste or their ammonia is excreted across their gills. So something in the region of 70% of their ammonia is excreted across their gills. And then the rest is lost in urine and feces. The good thing about being able to do this is that little to no energy expenditure is required because ammonia is the first waste product that's produced through the metabolism of proteins and nucleic acids. You don't have to invest any energy to convert ammonia into a less toxic product. You can just let that ammonia diffuse across your body, or you can excrete it in some other way. And then because you're living in water, that ammonia will just dissolve into the water, and then it will be so diluted that you won't be exposed to it. If you're an aquatic animal, you can excrete your waste as ammonia. Doesn't require any energy or very little energy, and it will just sort of dilute into the water and it will be gone. Most terrestrial mammals and many marine vertebrates will convert their ammonia into a less toxic product called urea. So urea is less toxic than ammonia. We produce it in our liver, and once it's been produced in the liver, the circulatory system will carry it to the kidneys, where it is filtered out and then excreted in our urine. The downsides to this are the conversion of ammonia to urea requires the expenditure of quite a lot of energy. So we have to invest a lot of ATP. We also still need to use a certain amount of water. So urea is still toxic. It's still toxic to our tissues and to our cells, but it's way less toxic than ammonia. But it is still toxic. So we still need to dilute it in water so we can safely store it in our body before we can excrete it. Because obviously we're not living in water, so we can't just let it diffuse across our bodies. We have to dilute it in water, and then we store that waste product in our bladder, so in our urinary bladder, and then we excrete it. So we're using less water than if we were producing ammonia, but we still do need to use some water. So the benefits of urea is it's less toxic, so we can store it. The downsides to it, also the benefit is that we need less water than if we were using ammonia as a waste product. The main downside to urea is that it's energetically expensive to generate or to produce, and it also does still need some water for us to use urea as a waste product. Now, the reason I've got the shark there, the great white shark, is they actually do something quite interesting. So I mentioned previously that most marine fish will osmoregulate by drinking seawater and actively transporting salts out of the body and producing small amounts of highly concentrated urine. Well, a lot of sharks actually do something very different. They will convert some of the ammonia that they produce. They will invest energy to convert that into urea, and then they will store that urea or keep that urea within their body tissues. And what that does is it increases the osmolarity of their tissues, effectively making them isoosmotic or isotonic to seawater. So by storing that or keeping that urea within their body, they are reducing the amount of water they lose through osmosis and the amount of salts they gain through diffusion. The downside to this is that, as previously stated, urea is still pretty toxic. So to be able to maintain these high levels of urea within their tissues, they have to have special molecules that they produce that actually detoxify that urea. but it's a bit of an odd one. It puts sharks in a bit of a strange position where people will often refer to them as being osmoconformers. But essentially, they're still osmoregulators because they're having to invest energy to convert ammonia into urea so they can keep that urea within their tissues to make them isoosmotic. Okay, so it's a different approach to doing the whole thing. The third type of nitrogenous waste is the uric acid. And uric acid is produced by insects, land snails, birds, and many reptiles. The benefits of uric acid is that it is relatively non-toxic and it also does not dissolve readily in water. And we tend to see it produced or excreted as a paste with very little water loss. So this is great if you are an animal that does not have access to lots of water. So we start to think about lots of reptiles that might be living in hot environments, or we're thinking about insects and certain snails that might either not have access to water or they may lose a lot of water through evaporation. And birds, well, birds don't necessarily have those problems. The problem that birds have is water is very heavy. So one of the adaptations that birds have to enable them to fly is that they don't have a urinary bladder. Because to have a bladder, you'll store water. Water is really heavy, and that will cost you more energy to fly. So instead, they invest energy to convert ammonia into uric acid. That means that they don't have to dissolve it in water. They don't need to then have a urinary bladder, so they can maintain their lightness for flying. So again, though, that comes at a cost. Uric acid is more energetically expensive to produce even than urea. So uric acid is the most costly of the three to actually produce, but it is also the least toxic of the three, and it requires the less water. So uric acid is great for water saving, it's non-toxic, but is the most energetically expensive to produce. And here we can see a summary of those three slides on this one diagram. So we have proteins and nucleic acids that get broken down into the amino acids and nitrogenous bases that contain the nitrogen. And then here we can see the amino groups. Here we can see most aquatic animals, including bony fish, are just excreting their nitrogenous waste as ammonia. You can see there, NH3. Mammals, most amphibians, sharks, and some bony fish will actually invest a bit of energy to convert that ammonia into urea, and then they will excrete that. Many reptiles and birds, insects and land snails will go a step further, spend even more energy to convert their ammonia into uric acid, and then they will excrete that as a paste. Okay, so different animals will excrete nitrogenous waste in different forms. So now that we know that different animals will excrete their nitrogenous waste in different ways, let's talk a little bit about how that excretory process is carried out. So excretion is an essential part of homeostasis. Well, we've established that waste products continue to build up, so we need to excrete them to help maintain the osmolarity of our tissues. Most excretory systems produce urine by refining a filtrate derived from body fluids. So when we look at an excretory system, there are four main things going on there. The first thing is filtration. So this is filtering of body fluids. What we then notice, or when we'll look at this in a minute, we'll see that this filtration process tends to be non-selective. So it will just filter out pretty much everything from our body fluids. Not quite everything, but we'll talk a bit about that in a minute. But it's a non-selective process. That means that we then need to go through a process of reabsorption. We need to reclaim the valuable solutes that we didn't want to filter out. We can then go through a process of secretion, which is adding non-essential solutes and wastes to the filtrate. So anything else that was missed or that we didn't get rid of the first time, we can secrete back into the filtrate that's being produced. And then ultimately, we go through the process of excretion. So processed filtrate now containing nitrogenous wastes and other things, so drugs and toxins, for instance. This will be then released from the body. So it doesn't matter whether you're looking at a fish or a whale or a human. These are the four things that are going on, or the four key functions of excretory systems: filtration, reabsorption, secretion, and then ultimately excretion. So let's go through and investigate that a little bit more by looking at the mammalian system in particular. So as stated previously, when we're looking at vertebrates and talking about excretion, we're essentially talking about kidneys. So kidneys are the excretory organs of vertebrates, and they function both in excretion and in turn osmoregulation as well. And we've got numerous tubules in the kidneys, and they are highly organized. So a bit like when we looked at on the previous slide, when we looked at the nasal salt glands of the albatross, you have this specialized arrangement of tubules that rely on counter-current flow and this specialized arrangement to produce this secretory fluid, or the salt fluid in the nasal glands. We see similar kinds of arrangements within our kidneys. They're very, very complex arrangements. So the vertebrate excretory system will also include ducts and other structures that carry urine from the tubules out of the kidney and ultimately out of the body. So we can see that on the left part of this diagram. So we can see here that we've got a pair of kidneys, and the kidneys have a renal artery that delivers blood to them, and blood is removed via the renal vein. The urine is produced within the kidneys. So we can see here the close-up of the kidney. We have the renal cortex along the outer edge here, moving into the renal medulla or the renal medulla, depending on how you like to pronounce it. Here we can see the artery that's delivering blood into the kidney, and then here's the renal vein. And then we can look closer up, and what you'll see is this arrangement of tubules all the way through the kidneys. And these are called nephrons. So nephrons are these tubules, these special arrangements of tubules that actually carry out those four processes that we talked about just now. Or specifically, they're carrying out the filtration, the reabsorption and secretion, and then preparing the urine. And then what you can see over here is the urine that is produced within the kidneys will then move down from the kidneys via the ureters into the bladder, and then from the bladder the urine is excreted through the urethra. What we can see here is a close-up of one specific nephron, and I've got some animations to show you on the next 3 slides that will go through the 3 main sections of a nephron and talk to you a little bit about what's going on, but I'll quickly run you through it. So what we can see up here, you can imagine now that we've got blood coming into a nephron. So it's delivered to the kidney via the renal artery. And then it gets separated out into smaller and smaller capillaries. And then you end up with these networks of capillaries that are referred to as a glomerulus. Okay, and that's very, very thin, and it relies on blood pressure to filter out the contents of the blood. Blood is filtered through the glomerulus, producing a filtrate which is collected in Bowman's capsule. It then moves through the proximal tubule, down the descending loop, and then up the ascending loop. So you've got the loop of Henle. It then moves into the distal tubule, and then down the collecting duct, out into the ureters, down into the bladder, and then ultimately excreted out of the urethra. So let's go through and have a look at those animations, and they will walk through what's happening specifically at each of these different stages.
Nephrons regulate the composition of blood by a combination of 3 processes that transfer materials between the nephron tubules and the capillaries that serve them: filtration, secretion, and reabsorption. A nephron tubule and the blood vessels surrounding it have been straightened and simplified in this diagram. The gradation of color, lighter on top, darker on the bottom, corresponds to an increasing concentration of solutes in the interstitial fluid surrounding the nephron toward the center of the kidney. As blood flows through the glomerulus, a knot of porous capillaries, water and virtually all other molecules small enough are forced by blood pressure out of the glomerulus and into Bowman's capsule. This process is called filtration, and the fluid that accumulates in Bowman's capsule is called filtrate. Blood contains blood cells, plasma proteins, water, sodium chloride, hydrogen ions, bicarbonate ions, urea, glucose, amino acids, and possibly some drugs and poisons. Blood cells and plasma proteins stay in the blood. They are just too big, but all the other substances are filtered from the blood and form the filtrate that accumulates in the nephron tubule. As the filtrate moves along the proximal tubule, certain substances are transported from the blood and added to the filtrate, a process called secretion. Secretion of hydrogen ions helps to regulate body pH. Certain poisons are also secreted to remove them from the blood. This list summarizes substances contained in the filtrate. Filtration is not selective, so some of these substances are useful to the body, and it is important that they be returned to the blood. The kidneys generally need to recover water, sodium chloride, bicarbonate ions, and nutrients such as glucose and amino acids. This reclamation of valuable solutes and water is called reabsorption. Let's look at the process of reabsorption as the filtrate continues its journey through the nephron tubule. In the proximal tubule, reabsorption of bicarbonate ions helps regulate the blood's pH. Sodium chloride, glucose, and amino acids are actively transported out of the filtrate. This leaves the filtrate more dilute than the surrounding interstitial fluid, so water follows by osmosis. These solutes and water re-enter the blood. As the filtrate moves down the loop of Henle, the concentration of solutes increases in the interstitial fluid surrounding the nephron tubule. Since this portion of the tubule is permeable to water, water leaves by osmosis and is reabsorbed into the blood. This concentrates the filtrate. In the ascending loop, the surrounding fluid becomes more dilute. This portion of the loop is impermeable to water but not sodium chloride. Sodium chloride diffuses out, lowering the solute concentration of the filtrate and adding to the solute concentration of the surrounding fluid. Near the top of the loop, sodium chloride is actively transported out, further diluting the filtrate. More substances are reabsorbed from the distal tubule. Sodium chloride is actively transported out of the filtrate. Bicarbonate ions may be reabsorbed too, helping to regulate body pH. Some drugs and poisons are secreted from the blood into the filtrate at this point. And this is another place where hydrogen ions may also be secreted into the filtrate to further adjust pH. There is a lot going on here. The main point is that the nephron is able to expel unneeded or harmful substances from the blood by filtration and secretion into the filtrate, and the nephron can reabsorb substances useful to the body. The filtrate enters the collecting duct. As it moves down the collecting duct, it passes through a region where the surrounding fluid has a higher and higher solute concentration. Water leaves the collecting duct by osmosis, concentrating the remaining filtrate into urine. Urea is one of the main body wastes in urine, but here some urea actually diffuses out of the filtrate. The urea performs a useful function by adding to the solute concentration of the interstitial fluid and causing even more water to be reabsorbed from the filtrate. Even though most of the water has been reabsorbed from the filtrate, urine is still 95% water. The most abundant solute is urea, along with other nitrogenous wastes. There is some sodium chloride and traces of other ions such as calcium and potassium. Hydrogen ions make urine acidic, and drugs and toxins may also be present.
Okay, so that brings us to the end of this session. So hopefully that was useful, that run through of the animations of the nephron. I think they said in the animation there is quite a lot going on, so might be something worth watching a few times just to get your head around it a little bit. But as always, we'll finish this session off with a quick recap quiz. So I have 5 questions here. I'll read through the questions. I'll give you 5 minutes to answer them, and then we'll go through the answers together. So question number 1, what is osmoregulation? Number 2, what does hyperosmotic mean? Number 3, what does stenohaline mean? Number 4, why would animals excrete uric acid instead of ammonia? Number 5, what are the 4 processes of most excretory systems? Okay, 5 minutes to answer them, and then we'll go through the answers together. Okay, welcome back. Hopefully you've had a chance to answer those. Let's go through the answers. So question number 1, what is osmoregulation? Well, osmoregulation controls solute concentrations and balances water gain and loss. Now there's a lot of ways that you could have answered that question, but essentially what I was looking for was the fact that osmoregulation balances the water and solute concentrations within an animal. So as long as you've got something along those lines, you'll be fine. Number 2, what does hyperosmotic mean? Well, it means more concentrated, or a higher solute concentration. Number 3, what does stenohaline mean? Well, it means intolerant of fluctuations in salinity. And number 4, why would animals excrete uric acid over ammonia? Well, it is non-toxic and does not require much water, so is ideal for saving weight and also conserving water. Obviously, the downside to it is it's the most energetically expensive waste product to produce. And then number 5, what are the 4 processes of most excretory systems? Well, it's filtration, reabsorption, secretion, and excretion. So those are the 4 that we just talked about with the kidneys. So hopefully you did okay with those. But as always, if you got a few wrong, don't worry too much at this stage. You can use that to identify the areas in which you need to go back over and increase your knowledge. So that brings us to the end of this session then. So we've covered quite a lot in this session through looking at what osmoregulation is, how different types of animals do it in different types of habitats. We've talked about nitrogenous waste and its excretion, why it's important and how different animals do it and why. And then we finished off by looking at the vertebrate kidney. So a lot to go through. You might need to go over this a few more times to help it sink in. But hopefully you should be able to describe the role of osmoregulation in homeostasis. You should be able to discuss the osmoregulatory challenges faced by animals living in different habitats and how they are met. Describe the role of excretion in osmoregulation, and then describe the structure and function of vertebrate kidneys. Okay, so there's a lot there, but hopefully this was a decent introduction to those topics, and hopefully it was useful, and I will see you in the next session. Thanks very much.